Hello guys, and welcome back to another video tutorial. The last lecture of the topic of mutations from the chapter Principle of Inheritance and Variation. In this chapter, in this last topic, we are going to talk about the last type of mutations, that is referred to as gene or point mutation. So what are these gene or what are these point mutations? And then we are going to categorize this gene or point mutation into several different types. There could be substitutional mutation or frame shift mutation. So let me initiate this lecture and let us go with gene and point mutation and finish it out, as well as we will have to understand the different types of mutagenic agent, and we'll be completing this particular topic from your syllabus. So please look forward. You are going to focus here, that is about the gene or point mutation. What are the gene mutations, what are the point mutation, how does gene mutation or how does point mutation takes place in a cell. Please be focused here in a particular point, that is gene or point mutation. So we are going to talk about the gene or point mutation. So see, gene or point mutation, the mutations that influences the expression of a gene, by altering a particular nucleotide, when a particular nucleotide is being altered, then such type of mutations are called as gene or point mutation. Then such type of mutations are called as gene or point mutation. A particular nucleotide is being altered. Now, such type of mutation can be of two different types: the substitution or the frame shift mutation. These can be of two different types: the substitution or the frame shift mutation. Now, what do you mean by substitution mutation? Now, in the case of substitutional mutation, a particular nucleotide will be replaced by another nucleotide. When a particular nucleotide is being replaced by another nucleotide, then such type of mutations are said to be the substitutional mutations. When a nucleotide of a gene is replaced by another nucleotide, the mutation is referred to as substitutional mutations. Such type of mutations are said to be the substitutional mutation. When a particular nucleotide is being replaced by another type of a nucleotide, then such type of mutations are referred to as the substitutional mutation. Now, which are the different types of substitutional mutation? How does the substitutional mutation takes place? It could be even a spontaneous, that is inborn error of metabolism, or it could be caused due to tautomerization, ionization, or base pair analog. So let us focus upon the different types of substitution mutation, then we will talk about the different types of frame shift mutation. Okay, so please look here. You can just focus here. On your screen you would be able to find the different types of substitutional mutations. Those substitutional mutations are transitional substitution mutations, including tautomerization, ionization, and base pair analog, as well as transversion substitutional mutation. The transversion substitutional mutation in the presence of methyl methane sulfate and ethyl methane sulfate. So we are going to talk one by one about each one of these. हम एक एक करके इनके बारे में पढ़ने वाले हैं. The first one being the transitional substitution mutation. Now, what are these transitional substitution mutation? How they are being caused? What causes transition substitutional mutation? So let us focus upon this A part, transitional substitutional mutations. Okay. So here we are. We are going to talk about transitional substitutive mutation. transitional substitution mutations. Now, in the case of transitional substitution mutation, the first type being the tautomerization. What do you mean by tautomerization? A. The first type being tautomerization. Tautomerization. Tautomerization in chemistry, you would have understood or you would have heard about this word tautomerization. The shift in the position of hydrogen atom across a double bond is called as tautomerization. It refers to the shift in the position of the hydrogen atom across a double bond. So if there is a double bond and across the double bond the shift in the position of the hydrogen atom is being made, then it is called as tautomerization. The types of tautomerization that are very, very commonly known as the immunotautomerization or keto-enol tautomerization. Such type of tautomerization takes place in the presence of a chemical agent such as nitrous acid. For example, the amino group containing nitrogenous bases such as adenine, which is also referred to as 6-aminopurine or cytosine, adenine or cytosine, in the presence of nitrous acid can transform their amino group into imino group. In the presence of nitrous acid gets transformed into NH, imino group. This is amino group into imino group containing nitrogenous bases, imino group containing adenine and cytosine, and these imino group containing compounds will perform forbidden base pairing. Such type of compounds will perform forbidden base pairing. So you can look here that these compounds will have imino group, thus performs forbidden base pairing. Okay, so this is the forbidden base pairing that we usually find. Now there is the tautomerization. This is immunotautomerization. This is immunotautomerization. There could be another tautomerization that could take place, and that is referred to as the keto-enol tautomerization. The keto-enol tautomerization. Now, how does the keto-enol tautomerization can take place? That we have to understand. In the case of keto-enol tautomerization, the keto-guanine or thymine gets converted into their enol group and performs forbidden base pairing. In such a case, the adenine which was binding with thymine will now bind. This adenine will not bind with thymine, but this will perform forbidden base pairing and will bind with cytosine. So this is referred to as tautomerization. The other type is, instead of amino, we will be talking about keto-enol tautomerization. In the case of keto-enol tautomerization, what is going to happen? Keto-enol tautomerization. The keto group of the nitrogenous bases. Is going to be converted into another group, that is enol group. Now how does it happen? The keto-guanine or thymine gets converted into the enol group. Keto-guanine or thymine gets converted into the enol group. and again perform forbidden base pairing. In such type of base pairing, what is going to happen? Then again forbidden base pairing is going to take place. The thymine, which was binding to adenine, is now going to bind with another guanine. So this is how the formation of keto-enol tautomerization influencing the formation of mutation. देखिए कीटो-एनॉल टॉटोमराइजेशन या अमीनो-इमिनो टॉटोमराइजेशन दोनों ही ऐसे टॉटोमराइजेशन हैं जो स्टेबल कीटो फॉर्म को इनॉल फॉर्म में, अमीनो फॉर्म को इमिनो फॉर्म में कन्वर्ट कर देते हैं एंड देन दे परफॉर्म फॉरबिडन बेस पेयरिंग. फिर ये गलत बेस पेयरिंग परफॉर्म करते हैं एंड दैट इज व्हाई देयर इज अ म्यूटेशन दैट इज टेकिंग प्लेस. ओके. Now, another type of mutation that we are going to talk about after tautomerization is ionization. Now, how does ionization takes place? Ionization refers to as the formation of ions from the nitrogenous bases that can occur in the presence of certain agents, certain radiations that can cause the ionization of the nitrogenous bases. Ionization. It refers to the formation of ion. It refers to the formation of ions from the nitrogenous bases. From the nitrogenous bases. This ionization can take place in the presence of certain radiation, and those radiations are ionizing radiation and non-ionizing radiation. Okay, so there can be two different types of radiation, ionizing, non-ionizing. Here we are talking about ionizing radiation. Ionizing radiations including alpha ray, beta ray, gamma ray or X-rays can have clustogenic effect over the DNA. Now what does this word mean? Clustogenic effect. Clustogenic effect means they will break the phosphodiester backbone of the DNA. If this is the double stranded structure of the DNA, 5 prime to 3 prime, 5 prime to 3 prime, then under these ionizing radiations, what can happen? Certain nitrogenous bases can be removed. Okay, there are certain places from which nitrogenous bases have been removed. Nitrogenous bases have been removed. This is what is going to happen. The UV radiation causes thymine dimerization in such a condition, but that we are going to talk about in the topic of radiations. Okay, the third one is referred to as base pair analog. What do you mean by base pair analog? Base pair analog means that there is a nitrogenous base and its analog is being used, another constituent which could replace the nitrogenous base and can cause mutations, then that is referred to as the base analog. For example, you must have heard about 5-bromouracil, which is the base analog of thymine. 5-bromouracil is the base analog of thymine. Okay, so next we are going to talk about the base analogs. The third one, the base analog. The base analog refers to a nitrogenous base that refers to a chemical compound that can replace the nitrogenous bases. These refers to the chemical compounds that have the ability to replace nitrogenous bases and perform forbidden base pairing. forbidden base pairing. It can perform forbidden base pairing in the case of base pair analog. For example, we can talk about the 5-bromouracil, which is the base analog of thymine. For example, 5-bromouracil, 5-bromouracil, 5-bromouracil is the base analog of thymine. It is the base analog of thymine. That means in the presence of 5-bromouracil, it can perform forbidden hydrogen bonding at the place of thymine. For example, if there is a thymine, there is an adenine binding with thymine, then at its place in another cycle, 5-bromouracil will arise, 5-bromouracil, and will replace this adenine, A to T to C to G. Here would be guanine, which will bind with 5-bromouracil, and G will be bounded with C. Okay, so G will be bounded with C. So what is going to happen? See, when a purine is getting replaced with purine, or a pyrimidine is getting replaced with pyrimidine, then it is referred to as transitional substitution mutation. Or if a purine is being replaced with pyrimidine, or pyrimidine is being replaced with purine, then it is called as transversal substitution mutation. In the case of transition substitution mutation, what is going to happen? Purine is going to be replaced with another purine, or pyrimidine with another pyrimidine. Purine getting replaced with another purine, or pyrimidine getting replaced with another pyrimidine. In the case of transversion, the purine gets replaced with pyrimidine, or pyrimidine gets replaced with purine. This is referred to as transversion. So in this case, what is going to happen? The adenine, which is a purine, is getting replaced with another purine. So this adenine is getting replaced with guanine, purine replacing another purine. As one purine is replacing another purine, adenine and thymine is replaced by guanine and cytosine. So you can find here adenine and thymine has been replaced into guanine and cytosine, because adenine binds with 5-bromouracil, 5-bromouracil binds with guanine, and ultimately guanine and cytosine are being formed. So what you are going to get is adenine is replaced with guanine and thymine is getting replaced with cytosine. So purine getting replaced with purine, and pyrimidine getting replaced with pyrimidine. So that is why this is called as transitional substitutional mutation. Transitional substitutional mutations. Okay, so you can look forward, all of these are referred to as the transitional substitution mutations. Okay, now in our next topic that we are going to talk about, RF, we are going to talk about the transversional substitutional mutation. In the case of transversional substitution mutation, what is going to happen? The purine is going to be replaced with pyrimidine, and the pyrimidine is getting replaced with purine. We are going to replace pyrimidine with purine, or purine with pyrimidine. Now how we are going to perform the transversal substitutional mutation, that we have to visualize. So this transitional substitution mutation is over. The next one is transversion substitution mutation. The transversion substitution mutation. Transversion substitutional mutations. Okay. In the case of transversion substitutional mutations, the purine nitrogenous bases gets replaced with with pyrimidine and vice versa. That means pyrimidine gets replaced with purine. Then it is referred to as transversional substitutional mutation. Now, in the case of transversional substitutional mutation, there are certain chemical agents which are being involved. Which are these chemical agents? One is ethyl methane sulfate, another one is methyl methane sulfate, catalyzing pyrimidine to purine transversion or purine to pyrimidine transversion. Now, that we have to understand how this transversion is going to take place. The ethyl, for example, methyl methane sulfate, methyl methane sulfate, ethyl methane sulfate, ethyl methane sulfate. When the DNA is dividing, supposingly this DNA is being dividing, then ethyl methane sulfate or methyl methane sulfate arises at its new nucleotides at certain key positions. When it further divides, then they perform forbidden hydrogen base pairing. Performs forbidden base pairing. That is, both methyl methane sulfate and ethyl methane sulfate performs forbidden base pairing. When they perform forbidden base pairing, then what is going to happen? Then the thing that is going to happen is that a new nucleotide will be substituted. You can look here. A new nucleotide has been substituted. A new nucleotide, supposingly a new nucleotide in the presence of ethyl methane sulfate or methyl methane sulfate, a new nucleotide has been substituted. Now, if this new nucleotide is purine being replaced with purine, it is called as transition. If pyrimidine has been replaced with purine, it is called as transversion. So it could be of two different types, transition or transversion, depending upon which nitrogenous base has been replaced with whom. अगर प्यूरीन से रिप्लेस हो जाता है, जैसे इथाइल मीथेन सल्फेट मिथाइल मीथेन सल्फेट एक जगह पे आ गए डिवाइडिंग डीएनए के अंदर जब ये इंसर्ट होते हैं, आ जाते हैं, तो ये अपनी पोजीशन के सामने एक गलत न्यूक्लियोटाइड को एंटर करा देते हैं. जिस न्यूक्लियोटाइड को एंटर कराते हैं वो प्यूरीन भी हो सकता है, वो पिरिमिडीन भी हो सकता है. क्योंकि वो प्यूरीन हो सकता है और या वो पिरिमिडीन हो सकता है, तो उसी जगह पे जब ये अगर प्यूरीन को रिप्लेस कर देते हैं तो उसे हम क्या बोल देते हैं, उसे हम बोल देते हैं ट्रांजिशन. प्यूरीन को प्यूरीन से रिप्लेस किया, अगर प्यूरीन को पिरिमिडीन से या पिरिमिडीन को प्यूरीन से रिप्लेस किया तो उसे हम बोल देते हैं ट्रांसवर्जन. So this is all about the substitutional mutations. Now we are going to talk about the frame shift mutations. Which are the frame shift mutation? How does frame shift mutations takes place? So let us zoom out of this chart and move to our topic. Okay, so here we are. Sorry students, just a minute. Okay, so here we are. We have to talk about the frame shift mutations now. The different types of mutagenic agents, the chemical agents and the biological agents. Okay, so we will be talking about them. This is all over. So we will be talking about the frame shift mutations. Now what are frame shift mutation? How does frame shift mutations takes place in a cell? Now frame shift mutations are the mutation that alters the reading frame of the DNA. Frame shift mutations. Frame shift mutations. Such mutations have have the ability to alter the reading frame, of the DNA, so as to form non-functional proteins. Now there are non-functional proteins that will be produced. For example, there is a drug proflavin, which drug could induce frame shift mutation. Now, how does this drug induces a frame shift mutation? Supposingly there is a DNA 5' to 3', this being the template strand, another one being the encoding strand, again 5' to 3'. One of the strand is getting transcribed into an RNA, and then there are sequences AUG, GAG, GUG, and so on, till there is a termination codon UGA, UAG, or UGA, UAG, or UGG, open number or local. If there is a change in the nucleotide, for example GUG, which was coding for valine, for example GAG, which was coding for valine, has been replaced with a mutagen to GUG, AUG, GUG, so on, UGA. Now see this, GAG getting replaced with GUG, glutamate getting replaced with valine. GAG is coding for an amino acid that is called as glutamate, and GUG encodes for an amino acid that is called as valine. So glutamate is getting replaced with valine, then such type of protein will be non-functional. Protein becomes non-functional. Proteins becomes non-functional. Such type of protein completely becomes non-functional in nature. Okay, so such type of replacement is going to take place, glutamate to valine, protein becomes non-functional. This is the critical case of an anemia, which is commonly called as the sickle cell anemia. For example, it occurs in the case of sickle cell anemia. Now let us have a look at the classical case of sickle cell anemia. How does sickle cell anemia takes place? What happens in the case of sickle cell anemia? So we are going to talk about the diseases that are being caused by the gene mutations. Some of the diseases that are being caused by the gene mutations are as follows. Diseases caused due to gene mutations. Now, which are the diseases that are being caused due to gene mutation? The first disease being sickle cell anemia. Sickle cell anemia. In the case of sickle cell anemia, the RBCs alters their shape from a biconvex to sickle shaped. In this disease, RBCs alters their shape from a biconvex structure to sickle shaped. once they are sickle shaped, they loses their ability to transfer the oxygen molecules. They lost their ability. They loses their ability to transfer the oxygen molecules. Now what is happening at the molecular level? What is happening at the molecular level? Molecular level पे क्या हो रहा है, इस बात को समझने के लिए आप यहाँ पर स्ट्रक्चर देखेंगे. There is a DNA which is present. This is which chromosome? 11th chromosome, 6th position, where the glutamate is being replaced with valine. 11th chromosome, 6th position. Gene is beta globin gene. Beta globin gene. The glutamate is getting replaced with valine. That means at this place the GAG getting replaced with GUG. So what is getting replaced? The adenine is getting replaced with uracil. Adenine is getting replaced with uracil. When adenine is getting replaced with uracil, then the type of mutation would be transversion. Because the purine has been converted into pyrimidine, has been transformed into pyrimidine. Okay. So at certain key important point within the DNA, there is a mutation, so that in the place of GAG in the mRNA, the sequence is GUG. GAG was going to code for glutamate, but instead of glutamate, what would be produced? At this place there is a mutation, and what is present? GUG. So here we get an mRNA in which GUG is present. So at the place of glutamate there is valine. Now how does sickle cell anemia actually occurs? Sickle cell anemia occurs because when the glutamate is being replaced with valine, then what happens? Then there is a hydrophobic pocket that is being produced. Now that we can understand here, that how does it occur? This occurs by the replacement of a hydrophobic pocket, by the formation of a hydrophobic pocket. Now see this. This is a hemoglobin molecule. There are 2 alpha and there are 2 beta. Okay. Now when mutation occurs within the beta, when there is a mutation, then valine produces hydrophobic pocket. Hydrophobic pockets. This will cause this hemoglobin molecule, this hemoglobin to be bounded together. One hemoglobin molecule is going to bind with another hemoglobin molecule, and the other hemoglobin molecule will bind with third hemoglobin molecule, and then will fourth. This will cause the interconversion of a globular protein into a fibrous structure. ये क्या करेगा? ये एक हीमोग्लोबिन का मॉलिक्यूल दूसरे से बाइंड कर जाएगा, दूसरा तीसरे से बाइंड करेगा, तीसरे से बाइंड करेगा, तीसरा चौथे से बाइंड करेगा, और इस तरीके के सारे हीमोग्लोबिन के मॉलिक्यूल एक दूसरे के साथ में बाइंड कर जाएंगे, जिसकी वजह से एक ग्लोबुलर प्रोटीन, हीमोग्लोबिन इज अ ग्लोबुलर प्रोटीन, बिकॉज ऑफ फॉर्मेशन ऑफ हाइड्रोफोबिक पॉकेट्स, because of formation of these hydrophobic pockets, the hemoglobin gets converted into a fibrous structure from a globular one. Hydrophobic pockets, formation of fibrous hemoglobin structure from globular. ग्लोबुलर से ये फिब्रस स्ट्रक्चर बना देता है. क्या बना देता है? Fibrous hemoglobin structure. Once the fibrous hemoglobin structure is being formed, the RBCs became sickle shaped because of the fiber-like structures which are being produced within the structure of the RBCs. See, this is very important. No teacher is going to tell you anything about sickle cell anemia. आप किसी भी टीचर से पूछ सकते हैं सिकल सेल एनीमिया का मॉलिक्यूलर मैकेनिज्म नहीं बताएंगे वो केवल रटते हैं और बोल देते हैं ग्लूटामेट की जगह वेलिन आ गया. लेकिन सही मॉलिक्यूलर मैकेनिज्म क्या है? सही मॉलिक्यूलर मैकेनिज्म ये है कि ग्लूटामेट की जगह जब वेलिन आता है तो वो वेलिन क्या बना देता है? ग्लोबुलर हीमोग्लोबिन को किसमें कन्वर्ट कर देता है? फिब्रस हीमोग्लोबिन में. जैसे ही ग्लोबुलर से फिब्रस हीमोग्लोबिन बनता है, दिस इज फिब्रस हीमोग्लोबिन. फिब्रस हीमोग्लोबिन hence cannot bind with oxygen effectively and hence it produces a condition of anoxia. यानी कि हम अब रेस्पायर नहीं कर पाते हैं, बॉडी के अंदर ऑक्सीजन की कमी हो जाती है. इट लीड्स टू कंडीशन ऑफ एनॉक्सिया कंडीशन, लो ऑक्सीजन in the blood, headache, or can even lead to coma and death sometimes. Okay, such type of condition takes place. So this is an 11th chromosome set. So the sickle cell anemia is an autosomal disorder. Such type of RBCs are destroyed in spleen and so cannot complete their lifespan, That is why the condition is referred to as anemia. So anemia. Okay. So this is about sickle cell anemia. Another very important point about sickle cell anemia is that in the case of sickle cell anemia, the malaria parasite Plasmodium cannot complete its life cycle. That is why sickle cell anemic persons are resistant to malaria infection because of predetermined destruction of RBCs within the spleen. सिकल सेल एनीमिया इस बात के लिए भी जाना जाता है, जिन पर्सन्स को सिकल सेल एनीमिया होता है, they are completely resistant to malaria parasite Plasmodium. Why? Because Plasmodium requires the complete life cycle to be for its complete life cycle to be terminated. It requires the RBCs to remain alive for 120 days, but RBCs here are being destructed within spleen before completing their entire life cycle. अपनी लाइफ साइकिल पूरी नहीं कर पा रहे, उससे पहले तो वो स्प्लीन में डिस्ट्रॉय हो रहे हैं. तो क्योंकि वो स्प्लीन में पहले ही डिस्ट्रॉय हो रहे हैं, इसलिए प्लाज्मोडियम अपनी लाइफ साइकिल को पूरा नहीं कर पाता और पर्सन जो सिकल सेल एनीमिक होते हैं, दे आर रेजिस्टेंट टू मलेरिया पैरासाइट प्लाज्मोडियम इन्फेक्शन. द लास्ट पॉइंट एंड द मोस्ट इंपॉर्टेंट पॉइंट इज दैट sickle cell anemic persons are resistant to infection by the malaria parasite, plasmodium, as it cannot complete its life cycle within such individuals, as it is not able to complete its life cycle, Okay, this is all about sickle cell anemia. There are two other diseases that are remaining, so we are continuing our lecture and we will finish it off. The next one is referred to as the alkaptonuria. Now what happens in the case of alkaptonuria? We must understand. In the case of alkaptonuria, which is still an autosomal disorder, the tyrosine is getting converted in our body, normal metabolism. The tyrosine is getting converted into homogentasic acid. And in the case of alkaptonuria, homogentasic acid oxidase enzyme is absent. Alkaptonuria. There is a mutation in the genes coding the enzyme homogentasic acid oxidase. Homogentasic acid oxidase. This is the enzyme that is going to be absent. Homogentasic acid oxidase. Now, what happens in our body? In our body, the tyrosine is getting converted into homogentasic acid. In our body, in normal metabolism, the tyrosine is getting converted into homogentasic acid. Homogentasic acid. In the presence of the enzyme, homogentasic acid, and this homogentasic acid will get converted into acetoacetic acid. Acetoacetic acid. In the presence of this enzyme, that is homogentasic acid oxidase. Now, supposingly this enzyme has undergone mutation. This enzyme is mutated. Okay? Mutation. Then what is going to happen? If this enzyme has been mutated, then homogentasic acid will be excreted in urine. When this is going to be excreted in urine, it will cause the color change. Changes color of urine. To black, so called as black urine disease. Okay. Another disease that we must understand after alkaptonuria is phenylketonuria. In the case of phenylketonuria, phenylalanine hydroxylase enzyme is absent. Phenylketonuria. In the case of phenylketonuria, the phenylalanine hydroxylase enzyme is completely going to be absent. Okay? The disease is caused due to the deficiency of phenylalanine hydroxylase enzyme. Now, in this disease, the tyrosine in our body, normal metabolism, gets converted into phenylalanine. In the normal metabolism, the tyrosine gets converted into phenylalanine. But what happens in the case of phenylketonuria, when there is phenylalanine hydroxylase enzyme is absent, then this interconversion cannot take place. This interconversion is catalyzed by phenylalanine hydroxylase. If this enzyme becomes absent, if this enzyme is absent, if it is absent, then tyrosine began to accumulate. Phenylalanine getting converted into, sorry students, in this disease what is going to happen? Phenylalanine is going to get converted into tyrosine. In this disease, if phenylalanine hydroxylase enzyme is going to be absent, then the phenylalanine will not get converted into tyrosine, but will get converted into another compounds. Which are those compounds? It will get converted into phenylpyruvate, phenyl acetoacetate, or phenyl acids. These will accumulate in brain, causes mental retardation, in the sufferers. So there is a disease causing enzyme phenylalanine hydroxylase. In its absence, the phenylalanine is not getting converted into tyrosine. Phenylalanine is also an amino acid just like tyrosine. Now the basic difference between phenylalanine and tyrosine is that in the structure of phenylalanine, what you find is that this is the structure of amino acid. There is an hydroxy group present. Okay, this hydroxy group must be removed in order to form tyrosine. This is phenylalanine, the hydroxy group is present here. This is phenylalanine and this is tyrosine. Okay, this is just a hydrogen present here. So the basic difference is that of hydroxy group. This hydroxy group, when it's being added, then it gets converted into tyrosine. If this hydroxy group is not added, it will get converted into phenylpyruvate, phenyl acetoacetate, or phenyl acids, etc., which will accumulate in brain and will cause mental retardation. The last disease is albinism, in which the tyrosine do not get converted into melanin because of the absence of the enzyme tyrosinase. आखिरी डिसीज है अल्बिनिज्म, जिसके अंदर टायरोसिन मेलानिन में कन्वर्टेड नहीं हो पाता बिकॉज ऑफ एब्सेंस ऑफ एन एंजाइम कॉल्ड एज टायरोसिनेज. सो द लास्ट डिसीज इज अल्बिनिज्म, in which tyrosinase enzyme is absent. Because this enzyme is absent, so tyrosine cannot get converted into melanin. Tyrosine forms melanin in the presence of tyrosinase enzyme. If this enzyme is absent, it will cause albinism, which is also called as Surajmukhi in Hindi. In such a disease, what is going to happen? The person will be referred to as albino, completely whitish, and the person will not be able to survive in the presence of direct sunlight because of very less amount of melanin present in its body. क्योंकि पर्सन की बॉडी में बहुत कम मेलानिन प्रेजेंट होता है, इसीलिए पर्सन सनलाइट के डायरेक्ट प्रेजेंस में सर्वाइव नहीं कर पाएगा. ठीक है, द पर्सन इज कॉल्ड एज अल्बिनो. पर्सन is called as albino, usually suffers from skin cancer. This is being caused. Skin cancer is being caused in such type of individuals. So this is all over. So all the different types of diseases are being done. Now what we have to focus here is about the three different types of mutations that we are going to talk about. Three different causes of mutation. Causes of mutations. Which are the different causes of mutations? Causes of mutation. Mutations. Mutations are being caused because of the presence of a particular chemical agent called as mutagens. These are being caused. These are being caused in the presence of a chemical referred to as mutagen. Now, mutagen can be classified into three different types: the physical mutagen, the chemical, and the biological mutagens. So, first, physical mutagens. Mutagens have been classified into physical mutagen. Physical mutagens. These brings about a physical change within the structure of DNA. These brings about a change within the structure of DNA, a change within the structure of DNA. These include radiations such as radiations which could be classified into following two types: ionizing and another could be non-ionizing. Non-ionizing. The ionizing radiations could be alpha rays, beta rays, gamma rays. The non-ionizing radiations could be UV rays. Here under ionizing radiation, X-rays are also included. Causes cleavage of the DNA. They causes cleavage of the DNA. Causes DNA cleavage, so called as clustogenic agents. Whereas the UV rays causes TT dimerization, causes thymine dimerization, and hence mutations. Within the structure of the DNA. Then there could be other physical agents such as heat, radiations. Other could be heat, the temperature, pH, osmotic pressure, etc. All this heat and cold treatment, all these types of factors are the physical mutagens that may influence the structure of the DNA and can cause mutations. After physical mutagens, the other type of mutagens are the chemical mutagens. The second one being the chemical mutagens and the third one being the biological mutagens. The second is referred to as the chemical mutagens. These include different chemical compounds that can react with the structure of DNA and can mutate them. Include chemical compounds. That can vary the structure of DNA. It can vary the structure of DNA. Okay, then it is referred to as chemical mutagens. Now these chemical mutagens can be different types: the mustard gas, the formaldehyde, the carbon tetrachloride, or the other different types of chemical agents. These include, for example, we have just talked about methyl methane sulfate, ethyl methane sulfate, the nitrous acid, the formaldehyde, HCHO, etc. So all these are chemical agents that can cause mutations. Then there are some very well known chemical agents such as nitrous acid UV radiation that can convert one compound into another. For example, adenine can get converted into hypoxanthine in the presence of nitrous acid. In the presence of nitrous acid, the adenine can get converted into hypoxanthine in the presence of nitrous acid. Such type of mutations can take place. Such type of agents are called as chemical mutagens. The last one being the biological mutagens. The biological mutagens. The biological mutagens include viruses, bacteria, etc. that can alter, it is usually viruses that can alter the genetic composition or sequence of our DNA. Then such agents are called as biological mutagens. Then these are referred to as biological mutagens. For example, there are viruses such as avian leukosis virus, the simian virus 40, SV40 virus. Sometimes HIV, it can also cause cancer. HTLV, human T cell lymphotrophic virus. All of these have the ability to act as a biological mutagens and can lead to formation of cancer. Cells when exposed to these agents can lead to cancer due to mutations. So students, this is all about mutations. We have completed mutation in every possible manner, in every type of explanation that was possible. So this is Dr. Nitin Wahi, and I would like to request you all to send your queries at my WhatsApp number in the groups so that I could answer your queries. Your questions are there. You can go with your MCQ booklet. आप अपनी MCQ booklet लगाएं और उनसे questions मुझसे ज़रूर पूछें ताकि हम लोग एक healthy discussion करके और better तरीके से understand कर सकें. Now we will be starting from the chapter 1, okay, and then we will be continuing our talk. Okay, thank you. Thanks a lot and best of luck for your studies.